Hi everyone, welcome and thank you for being here to listen to my talk about cell painting with the Cell Insight platform, which is an automated high-content imaging and analysis system. So I'm Kalpana. I am the technical product specialist for high-content analysis with Thermo Fisher Scientific. So just briefly, one of the benefits of working with such a large company like Thermo Fisher Scientific is that we have so many products and other companies that we have integrated into our workflow to benefit you. So for example, Applied Biosystems, Invitrogen, Gibco if you need cell culture reagents, molecular probes for all of your dyes for imaging, and there's so many more products in this list on the left-hand side. So I wanted to just mention that some of you may be familiar already with Manny Paniagua, who is our general specialist and covers many of these products. So if you have any questions about any of these products, please reach out to either one of us. And so as I mentioned, I'm Kalpana, and I am the high content technical specialist here. So just to give you a brief overview of the timeline of our history, so high content actually was invented by a company called Cellomics back in 1996. And so this is a timeline of the product development out of Cellomics. And as you can see from this timeline, in 2005, Cellomics was acquired by Fisher Scientific, which was then acquired by Thermo Scientific in 2006. So really our platform is the first high content imaging and analysis system out there. And so we've had a significant amount of time to put into the product development to make this really the best platform out there for high content analysis. And importantly, you can see that we introduced confocal imaging around 2008, and we surpassed 1,000 peer-reviewed publications back around 2016. In 2020, we introduced EurekaScan, which I'll talk to you a little bit about. And then just earlier this year, about a month ago, we launched the new Cell Insight CX7 LZR Pro system. So I'll talk to you a little bit about that as well. So we, as I mentioned, we invented and patented high content technology. The first system was launched in 1998. And so we coined the term high content screening, which is used to describe automated imaging and analysis at a cellular level. So this was initially marketed towards the pharmaceutical drug screening market, but it has since expanded to also be beneficial for people like yourselves, academic researchers. We do have a benchtop platform as well that I'll describe to you. And since we have been in the market for so long, we've developed comprehensive field support. So we have technical specialists, field engineers, application scientists all over the country to help you with your needs, to help you get not only trained, but also to help you with assay development. So that's one of our biggest strengths, is that if you are starting a new experiment, a new biology, and you don't know what you're looking for, or you just want some advice on what probes to use or what markers to look for in your physiology, we can help you with all of that. And we'll help you develop the protocols in your software as well. We also have HCS 101 training, which are classroom-based trainings for people who own our systems or want to learn more about using the software. We will give you a 2 or 3 day training to really go in depth into the software and everything that it can do for you. And we do have the largest user base in the industry, with over 15,000 systems having been sold and over 2,000 peer-reviewed publications using our technology. So what is high content analysis? I've been mentioning these terms, high content imaging, high content analysis a lot, high content screening. So what is that? So basically it is a platform that allows scientists to understand phenotypic changes. And usually this is done in a population of cells in either a 96 well plate or a 384 well plate. We can use other types of sample holders, but these are the most common that I see used. And so our system does have a microscope in it. So it is microscopic imaging of cells. But the important part is that we have this algorithm, this AI-powered algorithm for automated analysis. So what that allows us to do is we can automatically image in 2 dimensions and 3 dimensions all of your cells. We can get down to subcellular image analysis. And all of this is automated, so we can quantify objects, we can quantify intensities, we can quantify anything that you wish to explore in your samples. And then we also have automated data and file management and organization. So these are very important, and I'll touch on that a little bit more later. And then we also have the automated results visualization. So if you want to pull up a graph of maybe intensity in your cytosol versus intensity in the nucleus, that is easily possible with our software. So as I mentioned, we have over 2,000 peer-reviewed publications using our technology, and we have over 40 different pre-built assays in our software. So as you can see, the left side of the screen, this is a screenshot of what our launching screen of our software looks like. So we have over 40, as I mentioned, different pre-built assays. So these are very accessible to biologists. So if you're, for example, studying autophagy, we have an autophagy assay pre-built into the system. And then on the right side, this is a list of different experiments that you can do using these assays. So pretty much anything that a biologist would want to study, we can do it. We can do anything from a basic intensity measurement or spot counting all the way up to very intensive, very involved neuronal profiling assay as well. And we do have the most powerful neuronal profiling assay on the market. So this is an example of kind of what those, we call them bio applications, those pre-built assays. So this is an example of what you could do with those bio applications. So for example, if you're studying heat shock proteins, you can image these and analyze these, and this is all done automatically. Oxidative stress, maybe you're studying autophagy, as I mentioned. We also have the ability to image samples in 3D, so not only 2-dimensional cell monolayers, but we can also look at, a lot of people use this for cancer studies, so if you're looking at organoids or spheroids, we can also image and analyze those. We can do neuronal profiling, synaptogenesis, as I mentioned before, so there's a lot that we can do with our system. And so the main benefit of our software being automated as it is that everything is done in real time. So a lot of our competitors will have you collect all of the images first and then do some post-processing to analyze and really get out that data that you're looking for. So the whole point of high content is to get to that data faster. So our system actually processes all of the data in parallel with image acquisition. So by the time it's done imaging your, for example, 96-well plate, you also have the data ready for you as well. So we can do this at several different levels. So we can, if you imagine like a Google Earth view, we can look at the zoomed out view of the whole Earth, which would equate to the plate level. So we can just look at what's happening overall on the plate. We can also drill down to the well level. So if you want to see what's happening at individual wells, maybe you've done a drug treatment and you wanted to compare which drugs are more effective, you could look at the well level. We can also get down to the field level. So if you're looking at multiple fields within each well, you could compare what's happening at the field level. And we can also drill down to the single cell level. So if you really wanted to see what's happening to the individual cells, we can also access that data. So this tool, it's a very useful technology for looking at your specific biology. So this is an example of just the amount of data that we can get out of the system in an automated, very quick fashion. So this is an example of a multiplex cellular toxicity assay, and it doesn't matter what cell type you're using. So we can count cell number, which is a very basic measurement. We can look at nuclear area, which is also very simple. We can also look at cell morphology. So for example, here we're looking at an example of hypertrophy. We can look at DNA damage, mitochondrial membrane potential, apoptosis. I mean, there's really no limit to what we can quantify in your cells. And then our software also, as I mentioned, provides all of this data in real time. So as the software is imaging your cells, you also see these numbers coming out of the system. And so that enables us to easily create these graphs, so you can really see what's happening in your cells. Here's another example of the benefits of using our system. It's a very versatile system. So the previous example, we were looking at two-dimensional cells. And in this example, we have hepatocytic spheroids. So we can also image very large samples. So these you can see pretty much take up almost the entire well in this 96 well plate. And so we can image these in great detail. We can multiplex. We can look at multiple different colors to label different structures in the spheroid. We can even look at some bright field imaging. And in this example, we're looking at mitochondrial function and looking at comparing those with the necrotic core of the spheroid as well. So we can look at this as we saw in the previous slide from the zoomed out Google Earth view, kind of, if you will. We can look at the plate view and see what's going on from that level. But we can also drill down and look at this from the cell level view. So one of the benefits of our software is that it allows you to cross-reference all of the data. So you have these automatically generated graphs that you can pull up after the imaging and analysis process is over. And you can look at, for example, here we're looking at spot average area versus spot average intensity. And so we can click on one of these spots within our scatter plot, and each of these corresponds to one single cell. So if we click on one of these spots, it will pull up that cutout of that individual cell for you to examine visually. So in this way, we can kind of do a quality check and also just look at what our images looked like and make sure that we're analyzing the data correctly. So if we look at these 4 spots towards the top with the highest intensity, you see that visually, yes, those do have very high spot intensity. And then if we click on a few of these spots towards the bottom with low intensity, you can see that, yes, indeed, those do have very low intensity in the 647 channel. So this is just one of the ways that our software is incredibly powerful. So now we're getting to the good stuff, cell painting, and this is why we're all here for this talk. So cell painting is the latest addition to our platform. It's a morphological profiling assay, and I'll get into a little bit more about the details about what this assay is. But this is the latest addition with our newest platform that we just launched. So what is cell painting? So as I mentioned, it's a morphological profiling assay. It is an immunofluorescence assay, so it does use fluorescent dyes, which are all available from Molecular Probes, which we worked very closely with. And we're able to identify and examine in 5 channels. We can examine over 1,500 morphological features of the cells by labeling different compartments. And our software is exceptional at being able to compartmentalize all of the different parts of the cell so that we can examine them using various treatments, such as drug treatments. So this is kind of how all of the data looks like when you get it off of the either the CX7 or the CX5, any of our platforms. This is kind of that zoomed out view of at the plate level. So you can just examine visually and look at your data. Or you can drill down and look at the field level if you want to look at some of your images and see how they turned out. And then in the cell painting assay, as I mentioned, we're labeling with 5 different channels. And so in this example, we're looking at the nucleus, we're looking at actin, the endoplasmic reticulum, the Golgi, cell plasma membrane, and the mitochondria as well. So you can look at each individual channel as well as the overlay of all of the different colors. So the cell painting assay, it's a multi-well comparison for drug treatment effects. So as I mentioned, we're using 5 different channels and we're profiling these. So what is the difference between profiling and screening? So you probably are familiar with high content screening or drug screening, where you're using maybe a 96 well plate and you have several different compounds that you want to screen, and you want to just compare the effectiveness of all of these treatments. So profiling is different in that you're really getting a whole profile of how the cell physiology responds to your treatments. So not just comparing whether a drug is effective or not, you're really examining what that does to the cell physiologically. And we're looking at, as I mentioned, several different compartments of the cell: the mitochondria, the ER, the Golgi, the nucleus. We're looking at how it affects the cell DNA. So we're profiling 1,500 different ways that the cell can respond to these treatments. And so how can we use this? What is this useful for? So this could be useful for profiling an unknown compound. Maybe you have a drug that you want to examine what it does to the cell, what's the mechanism of action, what is really the molecular profile of how this drug impacts the cells. We can also use this as a phenotypic compound profiling to find leads quicker. So if you know that a certain response is what you're looking for in the cells, you can really just screen out all of those drugs that give you that response that you're looking for. We can also reveal the biological function of unknown genes using this method, and several other uses for cell painting: disease modeling, drug repurposing. But really at its core, this is increasing the overall screening efficiency for scientists. So you're able to look at so many different ways that the cell responds and screen different treatments or different drugs, and get all of this data at a very high throughput fashion. So obviously we are doing this with our high content system, so everything is done automatically. So all of the data is also coming out with your images to increase speed and just your research efficiency. So we can also, again, look at this data at the cell level. And on the left side, I've just listed the different fluorophores that we're using for this assay. So we're looking at, again, the nucleus, nucleoli, ER, Golgi, the mitochondria, and seeing how all of these different parts of the cell are affected by whatever treatment you're using. And you can access all of this data at the cell level. We can look at these scatter plots that are automatically generated and click on individual cells to pull up those cutouts and examine them visually. And because we do work with molecular probes, we have this new ImageIt cell painting kit. So you don't need to individually seek out those fluorophores that you need for this assay. We've put them all together in a kit, which is available from the core store. So you can purchase this kit to do the cell painting assay. So imagine you're doing this cell painting assay, and you want to know how long does this take. It sounds like 1,500 different features of the cell. How are we supposed to get all of that data? So we've done this for you. We've figured out how long it takes to do some of these assays. So here we're looking at a 4-color assay using autofocus as well. And so in a 96 well plate, if we're just using the wide field mode and we want to scan the entire plate, this took us less than 3 minutes. And so you can imagine looking at one organoid or one spheroid per well, and how long would it take to image, not only image, but also analyze all of that data. So in under 3 minutes, you have your images as well as all of the quantitative data. If we wanted to do this same experiment in confocal mode to get a higher resolution of our cells or organoids using the same 4 colors with autofocusing, this took under 5 minutes. So again, that's under 5 minutes, not only to get your images, but also to analyze all of that data and have all of those quantitative details at the end. So I want to talk more about some of the other experiments that we can use with our system. So zebrafish toxicity, this is another big use for our system, and we have a lot of customers doing zebrafish experiments. So this is an example of zebrafish in a plate, and so it takes about 3 days for them to mature from embryos to this state. And as you can see, they're very large, and so we have the flexibility to image at several different magnifications. So we could go down to 4x, 2x, and now with the CX7 LZR Pro, we can even image these at 1.25x. So we can really zoom out and capture the entire zebrafish in one shot. So this is looking at the entire well of that plate in one image. And we could image these in 2 dimensions. We can also image these in 3 dimensions. So a lot of our customers are imaging zebrafish and looking at liver toxicity as kind of a surrogate for, or a precursor for mouse studies and looking at toxicity of certain compounds that could be used as therapeutics in the future. So we can image these in 3 dimensions, and then we also have the Amira software available, which was introduced a couple of years ago with our system. So you can render these in 3 dimensions, you can calculate volumes, you can really do a 3-dimensional analysis of how is my compound or how is my treatment affecting maybe the size of an organelle or the size of the liver, or how is this affecting any part of the physiology in 3 dimensions. Another very important tool that we have is the single molecule mRNA spot quantification. So we can do this using the ViewRNA assay. So ViewRNA is an assay that uses these RNA probes that are hybridized to different fluorophores. And we can use these to monitor what's going on at a genetic level, at the mRNA level in our cells. And because we are Thermo Fisher and molecular probes, we do have ViewRNA libraries that are pre-built, so you don't have to start from scratch here. You can use one of our pre-built libraries for this assay. And we can use our spot quantification tool to really analyze and quantify what's going on at a genetic level. So this is an example. We can see some spots here, and you can use our software to determine, whether these are cytosolic, are these nuclear, how big are they, how many are they, any kind of measurement that you would want to get out of this, we can do with our software. And again, we can look at these and drill down to the cell level details. So for example, if you see there's some cells with 9 spots per cell, you can just go and click on those data points and it will pull up the cell level cutouts and show you what those cells look like. If you wanted to click on a cell that has 1 spot per cell, you can click on that and pull up some images. So our software is very powerful. Our data management is really the best out there in terms of being able to just handle all of these large data sets and then cross-reference everything so that you are able to visualize not only the data, but also the images that correspond. I mentioned before that we have one of the most, or the most powerful neuroscience algorithms, the neuronal profiling. So I just wanted to briefly go over what we can do with that. So kind of how we used to do this before is we had our plate. We're looking really at the well level for each of those wells in our 96 well plate. And then we can drill down to the field level and look at neuronal cells. And so here are some sample images of what you could get from that. So our software enables us to really profile all of these cells. You can see some of the skeleton drawings. We can trace neurites, trace them back to their original cell bodies, and really profile what's going on with these neurites. So here's a publication, one of the first that came out from using our system. So they were profiling teratology in neuronal cells. And this is a more recent publication. Actually, two of my colleagues are co-authors, Nick Radio and Sesha Thakur, on this paper. So here they were looking at three-dimensional views of neurons. So not only can we do this in 2 dimensions, but we can also do neuronal profiling in 3 dimensions. So we acquire all of those images in 3D. And then, as I mentioned, we have Amira software, which can allow us to analyze all of this data in 3 dimensions. So you can see from this video, we used the confocal mode. We imaged through each of the different focal planes of the sample, and then we can use the Amira software to render and quantify everything in 3 dimensions. So this is incredibly useful if you have neuronal cells that are not just growing in one plane. I mean, they're not always just flat cells. So the neurites might be going in and out of that plane. And so we can capture the whole picture of what's happening with that neuronal cell. So now that I've talked to you about the different experiments that we can do with our system, I wanted to go over really what are some of the details of the technology that we offer. So we do have a few different systems that we offer. So the CX5, which you see on the top left, that is our kind of starter tool. That is a benchtop system. It has the wide field imaging mode, and the 5 in the name stands for 5 channels. So we have your standard 4 colors as well as a near infrared line. So this is a great tool for startup labs, or maybe you are just getting into the high content realm and you want kind of a starter instrument. We do also have the CX7 LZR platform and the newest Pro, which we just introduced about 1 month ago. So the CX7 platform is our confocal tool. So this has the capability of wide field imaging as well as confocal imaging. So all of our platforms come with this HCS Studio software, which I've shown you some screenshots of and talked to you a little bit about. So the best part of our software, as I mentioned, is that it's real-time analysis. So it's workflow driven, and it provides you with that real-time feedback on your assay so that you can make quicker decisions, you can get to your data faster. And all of our systems come with the store data and image management software. So what this does is you can imagine if you're using this system to run several plates per day, every day of the week, you're going to generate a lot of data, maybe terabytes of data. So you have images and you have the numbers associated with those images. So all of this data has to be managed some way. So our store data and image management software allows you to not have to think about that. So it gives you a centralized database where you could be using the instrument to run some assay, and then on your laptop, 2 rooms down, you're looking at your data from yesterday. So it allows us to catalog all of the data in a way that, as I also showed you previously, you can cross-reference everything. So once you have all of those images, you can pull up your scatter plots and just click on a data point and see what that cell looks like. And the newest system, the CX7 LZR Pro, actually we've introduced this new photometrics back illuminated scientific CMOS camera, and I am going to talk to you a little bit more about what that does for us. So the back illuminated SCMOS camera, this is really a state-of-the-art camera. We are the only platform on the market that has this advanced technology in our high content system. So all of our competitors are using front illuminated cameras. And so if you think of camera technology, if you think of the sensor kind of like your eye, where you have your iris and you are visualizing, and then your retina is all the way in the back of your eye, and that's where the sensor really is. So light has to pass through your entire eye before it gets to the retina, which is the sensor. So that's kind of how a camera sensor works, with all the electronics and wiring and everything on top of the sensor. So light has to pass through all of those things before, all of those elements, detector elements, before getting to the sensor. And so you can imagine that there's a lot of loss, and so that is inefficient at really capturing all of the light that's coming out of your sample. So the benefit of a back illuminated SCMOS camera is that the sensor is on top. So all of your light is hitting the sensor first. So you're efficiently capturing all the light because it doesn't have to pass through all of the detector elements first. So this camera that we're using offers a peak quantum efficiency of 95%. So that gives us really stunning image quality, and we're capturing more of your data and losing less. We also have now introduced the Olympus X-line objectives. So these are very high resolution objectives. And we also now have expanded our capability to whole well imaging using a 1.25x objective. So these two put together enable us to not only have stunning image quality, but also high resolution. So these enable us to just image and get through your samples faster, get to that data that you really want much faster, and also be able to provide publication quality images. And with all of these changes, we're able to access several applications that we weren't able to before, such as cell painting, which I described to you previously, as well as the AbCam Fireplex high throughput assay, which is an immunological profiling assay. So a little bit more details about our systems. As I mentioned, we do have the Olympus X-line objectives available, all the way from 1.25x up to 60x magnification. Our systems are all capable, not only the CX7 LZR Pro, but the CX5 as well, are all capable of live cell imaging. So if you want to do kinetic studies, we offer temperature, CO2, humidity control, so that you can do those kinetic studies. The CX7, specifically the confocal platform, has this dual pinhole spinning disk technology. So we offer either the 40 micron or 70 micron pinholes, depending on what kind of resolution you're looking for and what kind of light gathering ability you need. The CX7 also comes with this 5-color bright field module for histology, if that's something of interest to you. And then CX7, as the name says, has 7 colors available. So we offer this either in LEDs or laser, as the CX7 LZR stands for laser. And so we can go all the way from blue, from 385, all the way up to the near IR, almost 800 nanometer excitation wavelength. And as I mentioned, we have the Photometrics SCMOS camera with 95% quantum efficiency. We also offer a patented laser autofocus system. So this is really state-of-the-art laser autofocusing system that allows us to detect where that where the change is from the plastic or your glass dish and your media, and it can help you maintain focus in your sample. We also have software autofocus as well, which is really a superior software autofocus that enables us to find the highest level of contrast in your sample very quickly during your automated assays. So this is an example of some of the fluorophores that we can use. As I mentioned, we work very closely with molecular probes. So any fluorophore that you want to use, we can help you get that. So the CX7, as I mentioned, we have the blue, the 405, 400 nanometer range, 450. We also can access the near IR up to 800 nanometer excitation wavelengths. So this gives us a very broad range of fluorophores, which enables us to do multiplexing really well. So if you wanted to look at 6 different colors in your sample, you can definitely do that and get the most data the most efficiently out of your samples. So just a little bit more about the camera. So comparing with our previous system, if you look at this graph on the left-hand side, the light blue line at the top is showing our newest camera, the Prime BSI, with max quantum efficiency of 95%. And as you can see, we still get very, very good quantum efficiency across all wavelengths, not just at the peak. And if you look at that second purple line right below the light blue line, that is our previous generation camera in our previous system. So we've significantly improved that quantum efficiency gap across all wavelengths. So we have very effective imaging at the near IR as well as the blue range. And this just enables us to, like I mentioned, get better image quality and also image much faster. So how much faster can we image and analyze with our new system, the CX7 LZR Pro? So we did some studies comparing our previous generation system with the Pro. We used a 5-color assay with a very intensive analysis algorithm as well. So as I keep mentioning, our software analyzes all of the data in parallel with image acquisition. So we have a dedicated graphics card that allows us to do all of this processing in parallel. So we set up a 5-color assay with analysis using laser autofocus in confocal mode, and we compared our previous generation system to the Pro. So if we're looking at a 96 well plate, this took us about 17 minutes with our previous generation, and we cut this time down to 4.5 minutes with the Pro. So this is almost a 4-fold difference. So our Pro system, as you can see for the 384 well plate as well, the Pro is at least 4 times faster than our previous generation system. And we could further optimize this to be even more faster than the previous generation. So as I keep mentioning, we do all of our data analysis in parallel. So other companies out there will have you do this all in sequence. So you'll prepare your plates, you'll image your plates, then you have to handle those images, maybe export them to another software or transfer them to another workstation. And then you do your image processing and data handling, and then you can look at all of those data and review. So this might take hours to do everything in sequence, whereas we have the technology to do everything in parallel. So you prepare your assay, and as soon as you put it into the instrument, the imaging, the image handling, processing, data handling, all of that is done in parallel at the same time. So when your imaging is done, all of your data is also done and ready for you to review. So we've cut down the time to minutes, as I showed you on the previous screen. We also, because this is an automated system, we do work very closely with lab automation to help you automate this even further. So for example, if you're working in a screening lab and everyone in your lab is doing drug screening and you all generate 10, 20 plates per day, we have a robot that is able to handle all of this for you. We can barcode the plates, you can do multiple different types of experiments, and the robot can handle all of that, up to 80 plates at one time with the robot. We have a scheduling software. So let's say 5 different people in the lab want to use this. Each of those 5 people can put their plates into these stacks, and you can program, you can tell the robot which experiments belong to which set of plates, and it will go through and schedule everything and do everything automatically for you. And because lab automation is part of Thermo Fisher as well, this is another benefit of working with such a large company. You don't need to do any additional work to integrate anything. All of this is included with our software, very easily accessible with our software. So another thing that we added to our software about 2 years ago is the Eureka Scan Finder. It's a seek and find functionality. And I'm not going to read over all of this, but this is just here for your information. This allows us to, again, save time, because that's the point of high content, is to get to your data faster. So we can really save a lot of time using the Eureka Scan functionality. So what is Eureka Scan? So pre-Eureka scan, let's look at an example of a 96 well plate, and we're imaging using 10x magnification. So using these settings, we would need to acquire 25 different fields for each well if we wanted to cover the whole well. So you can see here in this example, we have one spheroid, very small spheroid per well. So if we want to make sure that we capture every single spheroid on this plate throughout the 96 well plate, we would need to acquire 25 images for each well to be able to capture that one single spheroid in each well. And so this would take almost 30 minutes to go through and image and analyze the whole plate before we had Eureka scan capability. Now with Eureka scan, this is a double pass functionality. So it enables us to go through at a low magnification first. So on the right-hand side, we're using one field per well at a 2x magnification. We can do this in wide field to just speed it up and make everything faster. And so we just take one image of each well. So that might take maybe 1 minute, 17 seconds, and to acquire every well. So using that, Eureka scan can then go through and detect where the spheroids are within the well. It can find those coordinates for you and automatically switch over to a higher magnification. So in this example, the 10x, and only go back to those coordinates where it detected a spheroid and take one 10x image in each well to capture that spheroid. So this total scan time is maybe 1 to 2 minutes compared to the 30 minutes if we had to go through an image at 10x every single field. So this is an example of the kind of images that you can obtain using Eureka scan. So we can do the low magnification scan using wide field perhaps, because we want to image as fast as possible just to detect where the spheroids are. And then we can switch to confocal mode for the higher magnification and get these high quality, high resolution images. We can either do one slice, or we can do these, we can do Z-stacks and get the whole 3D image of that spheroid. So this is a summary of just how much time you can save. So in the first pass, like I said, we did a 2x, just one field per well, and that took about 40 seconds. And then using the second pass, the 10x, we can pinpoint those coordinates and only take one image per well at 10x with high resolution, high magnification. So the total scan time for this was only 2 minutes compared to the 30 minutes it would take if you wanted to go through and image every field and every well. So that's all I have for you today, and now I'm happy to take your questions.